Hi students, welcome to Sunil's tutorial. I'm Sunil Midwani and today we'll be doing this chapter called as genetic basis of inheritance. Now, let's see the 7 traits in the pea plant that were selected by Mendel. 7 traits in pea plant selected by Mendel. Right? Let's see it in tabular form. Character, pairs of contrasting characters. Now you will have dominant character and recessive character. Now, the pairs of contrasting characters, Mendel selected 7 different characters. One was the seed shape. Right? Seed shape could be round, that is my phenotype, and the genotype for this was represented as SS or wrinkled. Which was represented as small ss. Then the second character was, that was the first character or the first trait. Then seed color. Seed color, yellow, phenotype, genotype YY, green, phenotype, genotype YY. Third character was flower color. Red is my phenotype, genotype is RR, and white, that is RR. The fourth character was pod shape, that is inflated, that was II, and constricted, that was small I, small I. Then you have color of the pod. Green, that was GG, and yellow, which was GG. Then you had position of the flower. Position of the flower was axillary, AA, or terminal, AA. And the next was height of the plant. Which was tall, that is TT, and dwarf, that was TT. So these were the 7 traits in the pea plant that were selected by Mendel. Fine. These were the traits that he selected for his experiment. Fine. Do we get this thing here? Next. Now, what are the symbols used in genetic? Symbols used in genetics. Right? parental generation. Parental generation is represented as P or F1. Sorry, P1, not F1, P1. Then you have first filial generation, that is represented as F1. Then you have second filial generation, which is represented as F2. Third filial generation, represented as F3. Right? Dominant character is always represented with capital letters. You could have capital T for tall, or you could have capital Y for yellow. Capital Y for yellow. Right? Then recessive character. Recessive character is small t for dwarf or small y for green. Right? Then female. The female plant would be represented as, that would be a symbol for female plant in genetics, and the male plant would be represented as, this would be my symbol for male plant. So these are my symbols used in genetics. Fine, do we get this in here? Now once we know the symbols that are used in genetics, now we can actually start with Mendel's experiment, right? Let's see what did Mendel's experiment say. Let's before we do that, we just do a couple of more definitions so that when we are doing the experiment we have no problem. I don't think in the last lecture I gave you the definition for emasculation. Emasculation definition? No. Yesterday? No. Emasculation. In the last lecture? No. I just explained to you what is emasculation. Emasculation is nothing but the removal of stamen well before anthesis. Emasculation is removal of stamen well before anthesis. What do I understand by anthesis? That is formation of pollen grain. So even before the pollen grains are formed, you are going to remove the stamen, right? Emasculation, it is done in bud condition. Fine, do we get this in here? Okay. Next, now we can actually start with Mendel's experiment. The step and the procedure of Mendel's experiment. Steps of Mendel's experiment. Steps of Mendel's experiment with suitable example. Now, first let's see the procedure of Mendel's experiment. Of Mendel's experiment. What he did was, Mendel was very methodical. Mendel was very methodical in his experiment. Now, in order to make sure that there is no mistake in his experiment, he used pure breeding plants. Now, I have already given you the definition of pure breeding plants, in which both the alleles are similar. They are homozygous. Fine. So he used pure breeding plants. Right. Now, pure breeding plants also, he wanted to make sure that there is no mistake in his experiment. So he used pure breeding plants which were bred true for at least 3 successive generations. Which were bred true for at least 3 successive generations. So he made sure that he had those plants which were true breed for at least 3 generations. Right? Now, when he started his studies, he made sure that he studied only one character at a time. Right? So he first studied in inheritance, or just one character at a time. He first studied inheritance. He first studied inheritance of just one character at a time. Right? Unlike others who considered the organism as a whole, he didn't consider the organism as a whole. He just took out one character and he would study only that one character. Then he studied 2 characters together, then 3 characters together, and that's how. Right? So first he finished the entire study on one character. Now, while he was doing the second character, he took the first character also into consideration. Right? Then he studied 2 characters together. And then 3. So that's how he did his studies. Next, we first think what was the method used by Mendel for his experiment, right? Then in his experiments, he would always keep the generations separate and maintain separate records of actual number of each type of offspring. So he knew which was the parental generation, which was the F1 generation, F2 generation, and F3 generations. He would keep the different generations separate. In his experiments, he always kept generations separate. He always kept generations separate. In his experiment, he always kept generations separate and also maintained separate records. Of the actual number, of each type of offspring. What was the actual number of each type of offspring? That is both quantitatively and qualitatively. He kept records up to 3 generations. And right? Then during crossing, pollen grains and ovules were considered as the important organ which formed the bridge of heredity. He said that in order to make sure that proper hereditary, or in order to transfer hereditary from one generation to another generation, it was the pollen grains and the ovules that were considered as important organs. I can say that during crossing, pollen grains and ovules were considered important organs which form the bridge of heredity. Right? Now, he always performed crossing experiments using 7 pairs of contrasting characters, but considered only 1 pair of contrasting character at a time. He would do crossing experiments he has done crossing experiments for 7 pairs. Now, I've already told you what are the 7 pairs of contrasting characters that he has selected. These are the 7 pairs of contrasting characters that he has selected, but at a time he would do experiments only on 1 character, right? He performed crossing experiments using 7 pairs of contrasting characters, but considered 1 pair at a time. 1 pair of contrasting character at a time. Then he conducted enough number of crosses and reciprocal crosses to eliminate chance factor. So he did the experiment so many times that he knew that the results were definite. It is just not coincidence because of which he is getting these results. So by doing the experiments multiple number of times, he eliminated the chance factor. He conducted enough number of crosses and reciprocal crosses to eliminate chance factor. Right? Now, he conducted the experiments in 3 steps. So this was the method that was used by Mendel. Right? Now, he conducted the experiment in 3 steps. He conducted the experiment in 3 steps. Now, what were the 3 steps? Step 1. Step 1 was the selection of parent and obtaining a pure line. See, what did I tell you? That he made sure that he had pure bred at least for 3 generations. Right? So, selection of parents and obtaining pure lines. That was his first step. The first thing he wanted to do was to find out which were the to get parents such that they were pure lines, for which it involved selecting a pair of parents with contrasting characters. It involved involved selecting parents with contrasting characters. It involves selecting parents with contrasting characters and obtain each parents in pure condition. And obtain each parent in pure condition. For example, let's assume that if he was considering the character height of the plant, in which case what he would do is, one parent that he would take would be tall, that would be pure tall, TT, the phenotype would be tall, and the other character would be dwarf, that would be TT, but he would make sure that the genotype is homozygous. That means both the AMs are similar, right? So that was the first thing that he did. He took cross between contrasting characters, same character but the contrasting character with pure line, right? That was his first step in the experiment. Second step, step 2. Step 2 was artificial cross of selected parents. Artificial cross of selected parents. Artificial cross of selected parents to raise F1 generation. Raise F1 generation. Now once the parents were selected, then it involved crossing the selected parents. One parent was used as the male plant to provide the pollen grains, while the other was the female plant to provide egg. It involved crossing the selected parents. It involved crossing the selected parents. One plant was used as a male parent. One plant was used as male parent, and one plant was used as a male parent, providing pollen grains. And the other plant was used as a female parent, and other was female parent. The other was a female parent providing egg. Fine, do we get this thing here? Now, in order to make sure that the cross was properly done. Now, remember when we were studying about the characteristics of pea plant, we said that they are self-pollinating. Now, I didn't want them to self-pollinate. Hence, emasculation was done. Right? So, emasculation was carried out. Emasculation. I hope you all remember what is emasculation. Emasculation is the removal of the stamen before the formation of pollen grains. Right? So emasculation was carried out to prevent self-pollination. Self-pollination and fertilization. So that there would be a proper cross. Right? Stigma of the emasculated flower was covered. Stigma of the emasculated. Now, if I remove the stamen, there would be no pollen grains, so there is no chance of self-pollination. But it is quite possible that from some adjoining plant, which I don't want, a pollen grain would come. Therefore, the stigma of the emasculated plant was kept covered. The stigma of the emasculated flower was covered by a separate bag. Was covered by separate bag to avoid entry of other foreign pollen grains. To avoid entry of other foreign pollen grains. Fine, do we get this in here? So thus we made sure that there would be a proper cross between the two plants that we desire, right? Pollen grains were dusted on the stigma of the female plant for the desired hybridization. Pollen grains were dusted. Pollen grains were dusted on the stigma of the female plant. On the stigma of the female plant. Pollen grains were dusted on the stigma of the female plant. Female plant as in the emasculated female plant for desired hybridization. Fine. Once the pollen grains were dusted, then the cross-pollinated flowers were again enclosed by a bag to avoid deposit of any other foreign pollen grain. I wanted to make sure that I have cross between these two plants only, right? So I can say that the cross-pollinated flowers, the cross-pollinated flowers were again enclosed by a bag. To avoid deposition of foreign pollen grains. Fine. So I would then get the seeds were obtained which represented the F1 generation. This flower would give rise to seed. So I would say that seeds is the hybrids were obtained. Seeds were obtained which represented F1 generation. They were recorded and again sown in the soil and allowed to flower. They were recorded and again sown in the soil and allowed to flower. Right? So that was the step 2. And then Mendel observed that F1 resembled one of the parents. Mendel observed that F1 resembled one of the parents. Right? That was step 2. Step 1, get the pure parent. Then do cross between pure parent. Take one plant to be the male plant, the other plant to be the female plant. The plant you want as a female plant, you cut the stamen even in the bud condition only. Cover the stigma so that no foreign pollen grains fall on it. Right? When you want to do the cross, remove the bag, dust the pollen grains on it and cover the flower again with the help of a pollen, cover the bag again with the help of a plastic bag. Then from the flower you would get the seed, sow the seed again, you would get new plant. Now the new plant, that is your F1 generation that you would get, would resemble either one of the parent. Either it would be tall or it would be short. Right? Now, step 3. Step 3. Selfing of F1 hybrid to raise F2 generation. Selfing of F1 hybrid to raise F2 generation. Selfing of F1 hybrid to raise F2 generation. Now after flowering, they were allowed to carry on with self-pollination. The F1 generation that I got, I would do self-pollination of the F1 generation. So after flowering, they were allowed, to carry on self-pollination. Right? Now, so I got F1 generation, I've done self-pollination of F1 generation. Now seeds were obtained and they represented F2 generation. Seeds were obtained and they represented F2 generation. Seeds were obtained and they represented F2 generation. Now, they were recorded and again sown in the soil. They were recorded and again sown in the soil. Right? Now, when these plants grew up, the characters for which they were studied were noted. When these plants grew up, the characters for which they were studied, was noted. That means if I wanted to know whether I would get a tall plant or a short plant, so I did a cross between a pure tall and a pure dwarf. Right? I got my F1 generation which would either be tall or dwarf. Then I did self-pollination of that plant and I have got a new plant. Right? Now, I wanted to find out whether it is going to be tall or dwarf. Right? So the character for which they were studied was noted and both quantitative and qualitative results were obtained. Fine. So that was the entire experiment he would do. He would do the entire experiment in 3 steps. Get the pure parent, make sure that the parent is pure. How would I make sure that the parent is pure? By taking a pure parent for 3 generations. Right? Then I would do a cross between contrasting characters of pure parents and get the F1 generation. Then the F1 generation was allowed to cross with itself, that is self-pollination, to get the F2 generation. Then find out what is the character that the F2 generation has. How many seeds of or how many plants of F2 generation has one character and how many plants of that have the other character. So both qualitative and quantitative results were obtained. Right? Thus, I'll give you an example. If the tall and dwarf characters tall and dwarf character were taken as parents. Tall and dwarf characters were taken as parents in the initial stage. That the parents of F1 then the F2 generation, then in the F2 generation tall and dwarf occurred in the ratio of 3 tall is to 1 dwarf, right? So that's how he got the whole experiment. So similarly, he conducted experiments with all 7 contrasting pairs. Fine, do we get this thing here? So do we understand Mendel's experiment? Right, we'll stop this here for the day. Thank you very much.